This has got to be the lowest attendance of the whole year. I see a pretty small percentage of people who are here, of the class who are here. Okay. Two things to start. One, I do have a review session scheduled. As I said, it will be 6:15 p.m. tonight, and that will be in ALS 4001. And yes, I will videotape it, assuming we have no problems, and I'll get it posted as quickly as I can, hopefully later this evening. Second, exam format. Several people have asked about that. So I went back and looked at the scores on the last exam and found that the matching actually were better than the fill in the blank. So I decided to keep the matching. So so the format will not be any different than the first exam. I can't go back to the format of last term, unfortunately. I don't have enough TAs to grade. As I said, we'd be here for another 6 weeks if I had to do that. All right, so that's given. I will say a little bit about the exam. I recognize that, excuse me, there's a lot of material here, okay? So the exam covers everything through Monday. And I tried to be very careful in picking highlights, that is the main points in writing this exam. I tried to be as gentle as I could, as I told someone earlier today, okay? But I do recognize there's a lot of material to know on this exam. My advice in preparing for the exam is, as I've been telling many people, write things down. I think students get the notion that I've read it 10 times, therefore I know it. And writing it really confirms that you know it, and being able to write it without looking at it really is the ultimate test, because you're not going to be looking at it when you're taking my exam. When you're writing things down, my advice is make flashcards. And I know a lot of you do that, but a lot of you don't do that. Make flashcards, because not only do you get the practice of writing it down, but then you have the option of quizzing yourself as you're going through and doing those flashcards. And I find that really is a very good help to learning when you're trying to put into your head an awful lot of information. So as I said, I really did try to hit the main points, not get picked up on, stuck too much on details with this, and hopefully that you will do well in this exam. Okay, so that's where we are. What I plan to do today is start the material for the final exam. So everything starting from this point forward will be on the final. It will not be on the exam on Friday. And there's not much on here what I'm going to talk about today, actually. And then what I will do is open it up to questions, so we can have sort of a mini review session here if you'd like to ask questions, and do that. Okay? So that's kind of my plan for the day. We'll probably finish early, unless you have questions for the whole period, in which case I'll stay and answer questions for that. Okay. So last time I got talking, I interrupted DNA repair. So what I want to do is go back and just remind you a little bit about these different types of DNA repair, and then finish that up with this one I didn't talk about here. So DNA repair, as I said last time, is very, very important for cells to maintain the genome. Okay? It's very, very important to do that. If you have damage to critical DNA, then you really have to, and you really want to, be able to either fix it or get the cell out of the way. And we'll talk about that when I finish the section on repair. So being able to fix it, which is the repair part of that, is very, very important. And cells have an amazing collection of systems to repair damage to DNA. They have an amazing collection of those. So obviously you've seen proofreading, and proofreading I won't go over again. Proofreading is what DNA polymerase does, whether it's a DNA polymerase 1 or it's a DNA polymerase 3 or a DNA polymerase 2, in E. coli, for example, or most of your DNA polymerases. Now, not all DNA polymerases do proofreading, and as I mentioned before, some of the notable exceptions are things like the reverse transcriptase we find in HIV, which doesn't do proofreading. It has a very high rate of error. And for the survival of the virus, that actually is a good thing. It helps the virus to evolve rapidly. It helps the virus to evade all kinds of ways that would otherwise eliminate it. And as a consequence, it works very well for the viral scheme. It doesn't work too well for the host that has the virus, unfortunately. Mismatch repair, just to refresh your memory on that. is something that involves the mut-S, mut-L, mut-H system. Those names come from the system in E. coli. And as I said, there's a very similar system that exists in our cells and actually has a similar nomenclature, but I won't go into that. The system operates by having a protein called mut-S that recognizes mismatches within the DNA and binding to them. Okay? So this is a really good protein for recognizing mismatches. It'll find GTs, it'll find GAs, it'll find CAs, etc. If it's mismatched, mut-S will find it. When it finds it, it binds, and the binding of mut-S, that is where it stops, is a signal for mut-L to bind to mut-S. So mut-L, as I said last time, is a protein whose main function is to simply bridge the gap between mut-S and the protein that actually does the bulk of the work, and that's mut-H. So after mut-L has bound to mut-S, then mut-H comes along and binds to mut-L. And like I said, you won't have to know the system whereby the cell can tell which is the error strand. But the cell does have a way of telling which strand has the error. And mut-H makes an incision and chews back to get rid of that error that's in there. Then DNA polymerase comes along, fills it in with hopefully the right nucleotide, and everything is taken care of. Now this system works primarily where the proofreading system has failed. The proofreading system is very, very good, okay? But the proofreading system itself makes mistakes occasionally. So this is an added insurance against mistakes that the proofreading system doesn't catch. Okay? You might say, well, how often does this happen? You know, how big of a problem is this? Well, if we look at an E. coli cell that replicates a genome of 6 million base pairs, and it's got a system that recognizes an error once every 10 million base pairs or so. Basically, every time the genome divides, there's approximately one error in it that doesn't get fixed. Well, I'd say that's not very much, but imagine that E. coli can do 1 billion replications overnight. Okay? We start seeing how we could have a lot of changes happening. So the more the system has built into it, error repair mechanisms, the more stable the genome is. Okay? We also see with that 1 billion replications overnight how a cell actually can have a rapid system of evolution. That cell that starts making mutations in critical proteins that allow it to evade an antibiotic, for example, errors really work there. They really help the cell to be able to evolve new ways of working against the things that would otherwise kill them. Okay? So that works an advantage in addition to the disadvantage of the instability of the genome. Okay. Nucleotide excision repair, I went over it fairly briefly last time. And I gave as an example a situation where we had the thymine dimers that are produced by exposure to ultraviolet radiation. And those thymine dimers are depicted here. You notice that it's the joining between two thymines on the same strand, not different strands, but on the same strand. So this would be a T, this would be a T, for example. This system will also work if we have, let's say, a DNA adduct. That is one of those chemicals that has physically bound to one of those bases. This system would allow the removal of that section, as we see here. This is nucleotide excision repair. Removal of that section followed by replacement, as we see here. This is a different system than the mut-S, mut-H, mut-L system. Okay? But nonetheless, it results in, if everything works properly base paired DNA at the end. All right? Now, you might say, well, how does the cell know to use this one or use the mut-S or mut whatever? Okay? And that's one area of active investigation. But not surprisingly, this guy is going to look different than simply a mismatched base. So the base mismatch repair system is not going to have very big changes associated with it. And so the mut-S is really only going to recognize those mismatched bases, whereas we're going to have some other grosser deformities that we're going to see here, and this system will be involved in that. And no, you don't need to know the names of the enzymes. That's not a critical thing here. Okay, well, what I didn't talk about last time, but I will talk about this time, is a system that's on the surface kind of like the nucleotide excision repair system, except for this system is called base excision repair. Base excision repair. Well, what's the difference between a nucleotide excision repair and a base excision repair? Well, it turns out in the end they're not that different. They really are different only in how they start. So here's an example. We see up here a G paired with a U. There's a couple ways we can have that happen. One is if the cell mistakenly uses DUTP instead of TTP, and we'll have a U in there. Okay? If we have a methyl cytosine and it becomes deaminated, it becomes a U. We'll talk a little bit more about that later when we talk about eukaryotes and regulation. But that methylated C, which is not uncommon in a eukaryotic DNA, chemically is unstable and can go to become a U. So there's a couple ways we could have a base pair like we see here. Well, again, we don't want to have U's in our DNA, so the cell has a way of removing them. And this removal system involves an enzyme called uracil. Actually, it's called uracil N-glycosidase. Your book calls it uracil DNA glycosidase. Either is acceptable to me. And what it does is, you'll notice the very first step is it clips only the base. It clips only the base. And that's why we call it base excision repair, because the first thing that is removed is that U that's there. Okay? After that, there are then other enzymes that come in and clip phosphodiester bonds. Okay? Phosphodiester bonds, so that this gets chewed away, and then DNA polymerase fills it in with the proper nucleotide. Okay? So in the end, it's not that different from nucleotide excision repair. However, the first step is distinctly different. The net result of that is, again, we have properly base paired sequences at the end after we've had that happen. Okay, questions about that before I move forward? We don't want to think about this until the next exam, right? I understand that. Okay. So, I want to say a little bit at this point about cancer. And some of the things I'll say here also relate to things like aging, although it's not listed on here. All right? So, cancer, of course, uncontrolled cell growth. And we know cancers arise from alteration, usually in some critical protein or some critical protein or enzyme that is involved in helping the cell to decide whether or not to divide. So there's a variety of things that might happen. So the more mutation that we have, the more likely we are to have alterations in these critical proteins that control things in the cell, and the more likely the cell will therefore have uncontrolled growth. So protecting against mutation, obviously in a multicellular organism, is very, very important, just like it is for any other organism. And when we look at certain types of cancers, what we discover, not surprisingly, is that some cancers are very much more prone to occur when damage to the DNA repair systems has happened. Okay? A really good example of this is called HNPCC. This is a cancer of the colon. It's a colon cancer. It is an inherited colon cancer. And when you analyze, Dr. Andrew Burmeyer, who is an expert in this field at Oregon State, when you analyze the enzymes involved, the proteins that are defective or mutated in this cancer, what you discover is that some of the proteins involved in these systems I was talking about up here actually are defective. So they're unable to handle some types of repair. And because they're unable to do some types of repair, the cell is much more likely to have mutation in going through the replication process. Okay, so we start to think then, of course, it's not surprising then that mutation is in fact going to be a factor in the tendency of a cell to become cancerous. Okay. Well, another gene that's very, very important and very, very interesting in this respect is called P53. And P53, I think, is one of the most interesting genes that occurs in any cell anywhere, to be honest with you. And P53 is what people refer to as the quality control gene of the cell. Quality control. Other people call it the master switch of the cell, because the cell cannot go through the cell cycle unless P53 gives the OK. P53 has to give the OK for the cell to proceed through that cell cycle, the G1S, G0 mitosis that we talked about. Okay? P53 is sitting at the top of that. It's the king, as it were. Well, how does it do its thing? Well, it turns out if we look at the cell cycle, let me just take you back to the cell cycle to show you. If we look at the cell cycle, we have G1 to S. S is where synthesis occurs, and S is where the DNA is going to be replicated. P53 acts right here at this point. Okay? It acts at the boundary between G1 and S. And what it's doing is checking to see if the DNA is ready for replication. So if something has happened during the mitosis process, if some damage has happened to the DNA since the last time it got replicated, or there's something that needs to be taken care of, P53 will step in. Now, the mechanisms by which it steps in are many. P53, for a relatively small protein, has many functions, many of which are still being researched. One of the things that it does is it is a transcription factor. It can bind to DNA at specific sequences and activate the transcription of specific genes. Some of those genes are involved in DNA repair. This makes sense, because if P53 gets a signal that there's something wrong with the DNA, one of the things that it wants to do is make sure that it tries to repair that damage. Okay? So, assuming we have damage, P53 stops everything. The cell cycle itself stops dead in its tracks and will not enter the S phase. P53 goes and activates some genes that are involved in DNA repair. And those DNA repair genes then go to the sites of the damage and try to repair it. Meanwhile, the cell is sitting here twiddling its thumbs, waiting to decide. It's kind of like when you're sitting on an airplane and they tell you there's a mechanical problem and you're at the mercy of the mechanic that is doing things on the plane. There's nothing you're going to do until the mechanic says it's okay to fly, right? So P53 is giving that final okay whether it's okay to fly or not. The DNA repair proteins give their best shot. If, in fact, they get the damage repaired, then P53 backs away from the picture and the cell cycle continues. The cell goes and replicates its DNA and prepares to do mitosis as it would normally do. On the other hand, if P53 senses that the repair has not happened properly, the DNA repair proteins have not been able to fix the damage, then P53 will induce a process known as apoptosis. Apoptosis. One of the most mispronounced words in all of biology. The second P is silent. Apoptosis. You don't say pneumonia, right? It's not apoptosis, it's apoptosis. Or apoptosis. Either is acceptable. All right. In any event, apoptosis is cellular suicide. That cell will commit suicide at that point. And it does so by activation of a series of proteases that start breaking down all the proteins and everything inside that cell. The cell commits suicide. And that's a really good thing, because you don't want that cell, if damage has happened and you haven't been able to repair it, you don't want that cell going on and proliferating that damage and becoming a cancer cell. This is a good way of protecting a multicellular organism against potential cancer-causing cells. It's not perfect, obviously, because cancers still make it through. But again, without this system, we can imagine there's some real problems. When we look at the different types of cancers that are out there, we discover something very interesting. One of the things that we discover is that many, many, many cancers have mutations in P53. It no longer functions. It's no longer providing that protective control that says commit suicide. And the cell thinks everything's fine. It goes along and it proliferates, thereby making the cancer and everything else be worse. Okay? So damage to p53 is really not something that's in the cell's interest, because if p53 doesn't function properly, that quality control step during the replication process doesn't occur. Okay, that's a mouthful. Questions about that? Jenny?
Do you see the cell suicide in single-cell organisms?
Do you see cellular suicide in single-cell organisms? I can't say definitively no, but you would predict that you would not expect to see that. And the reason is that in a single-celled organism, her question was, do you see cellular suicide in a single-celled organism? The reason you would not expect that is there's no damage to having an out-of-whack cell going out there. And in fact, in terms of evolution, for a single-celled organism, the more diversity you have and the more changes you have, probably the better off the cell is. So as far as I know, there are no such things. Now, there is a similar, the only related phenomenon that I can think of in single-celled organisms that is at least slightly related is something called quorum sensing. So there are some bacteria that basically send little signals out in the medium in which they're growing. And those little signals are chemicals. And the more bacteria there are, the more of those chemicals that there are. And so for certain bacteria, until there's enough of those chemicals, they won't go through and become, for example, pathogenic and do their thing until they know there's enough of them. And they get that from the signaling. So that's a way of slowing down and stopping until you get things to an appropriate number. That's the only thing I know of in prokaryotic cells that's equivalent to that. I should also point out that there's, and this, you don't need to know this, but I'll just tell you, I said there were 5 DNA polymerases, okay, in E. coli. And there's 2 of them that are not listed on there. One of them is known as the sloppier copier. Okay? The sloppier copier is a DNA polymerase. And you might imagine from its name that it's a sloppier copier because it doesn't do as much error checking and proofreading that's there. The sloppier copier is made when the bacterial cell has had significant damage to it or has had significant problems such that maybe there's some place in the DNA where it's hard to get around it. A regular DNA polymerase won't handle it, so the sloppier copier comes in and takes its best guess at what it's doing. That's kind of cool. Yeah?
I know that there are bacteria that have natural plasmids, which will cause, when the bacteria replicates, it will cause, if a daughter cell doesn't get the plasmid, it will die.
Mm-hmm. So what he's saying is that there are, there are actually a couple ways that can work. So he's talking about a plasmid where if a daughter cell doesn't get the plasmid in a bacterial cell, it can die. That would happen, there's a couple of schemes by which that can happen. One is if the plasmid is carrying a critical gene that's necessary for life, and that would be one. The other, is, of course, is if you have and this happens a lot in resistance to antibiotics, if the bacterial resistance to the antibiotic is carried on the plasmid, which is one of the ways that it frequently is carried, then the same sort of phenomenon happens, that the protection against the antibiotic is carried in this plasmid. If the plasmid doesn't get transmitted properly, then you will have that phenomenon. But that's not what I would call a cellular suicide. That's a survival mechanism that is built in. So, but they are, I guess in a sense, they are that related. So, yes, sir. Is the p53 gene always active? The p53 gene is always there. It depends on what our definition of active is. Okay? So it's active when it's stopping. It's also active when it's making it go, because what it's doing is it's properly checking, is it okay to go or not go? In that sense, yes, it is always active. Is it always checking? Yes, it is. Okay. So that's what I want to say. There's one more thing I want to say about mutation, and then I'll talk very briefly about recombination. So we hear about all kinds of things out there that cause cancer, right? If you eat meat, going to cause cancer, right? Or you drink Diet Coke, going to cause cancer, right? So how do we decide if something causes cancer, all right? Well, one of the ways that we test for cancer is we take the thing in question and we inject it into a bunch of organisms, and we ask the question, do those organisms produce more tumors than those that get the control that doesn't have this? Okay, so I inject Diet Coke into one, I inject water into, well, it sounds kind of disgusting, doesn't it? This has a better ending than this, okay? So, we inject Diet Coke into one, we inject water into the other one, and we say which group of animals has more tumors. Okay? Well, that's costly. All right? And the animals aren't too happy with it. Right? unless they like Diet Coke, in which case they probably just want to drink it or something. Okay. What you really want to know is this compound causing mutation? Because mutation, something that causes mutation, is going to be very much more prone to cause cancer. So what we want to have is a way of studying the tendency of a compound to cause mutation during DNA replication. Okay? So what I'm going to describe to you is a system. I'm going to describe to you is a system that allows us to very quickly and easily see if, in fact, a particular compound is what we call mutagenic. A mutagenic compound is something that favors mutation. Okay? It's a test called the Ames test. It was invented by a man named Bruce Ames. And it's actually surprisingly simple. Okay? So let's go back to our Diet Coke example. We say, okay, there's something in Diet Coke that is going to cause mutation. This is going to happen, therefore I don't want to put it in. I don't want to have Diet Coke out there for people to drink, et cetera, et cetera. So instead of going and injecting a bunch of mice, I take and say, well, maybe I can answer the question with bacteria. Nobody minds if you kill bacteria. Nobody minds if you make them eat something they don't want to eat, right? Okay? So what you do is you make two sets of plates. You have a plate that you grow bacteria on that has no Diet Coke in it, and you have a plate over here that has Diet Coke in it. Okay? So that's one part of the test. We have a plate that has none. We have a plate that has some. Okay? Now, we also take in those plates, we put in an antibiotic into each one. So let's say we put in ampicillin into both. So only cells that grow on ampicillin will be able to grow on these plates. Why do we care about that? Well, we care about that because we have a strain of bacteria that I'm growing over here in my laboratory, and it has a little plasmid in it that gives resistance to ampicillin, except for one thing. Okay? This plasmid has a perfect gene for giving resistance to ampicillin, except for it's got one base that's the wrong base. And because of that one base being the wrong base, cells that have this plasmid cannot grow on ampicillin. But if you change that one base, then it makes a functional gene so that now the cell can be resistant to ampicillin. Everybody follow me on this? So I got this culture bacteria sitting here. They've got a plasmid in them that would let them grow an ampicillin if only they had one single mutation. Now we think about these billions of replications, right? The more these guys replicate, the more likely one of them is going to have a mutation in that particular base. All right? So I got my mix. I take it and I spread it onto these two plates. One plate has ampicillin and no Diet Coke. The other plate has ampicillin plus Diet Coke. And I ask the question, which one has more bacteria? Okay? If I see, and I want to use significant numbers as well, but if I see, and this is a slight variation on this, but if I see, for example, that I have a plate where I have a significantly larger number of bacteria than I do on the untreated plate, I can say, whoa, there's something in Diet Coke that is mutagenic because it favored making a mutation that allowed these guys to grow. Well, why in the untreated do I see any at all? What's that? Well, I started with a mixture that had 1 billion bacteria in it already, right? So there was some natural mutation that was happening at a background level. I never have perfect replication. That's why I have to compare untreated to treated. Because if the treated have more, clearly in becoming a colony, they had to have more opportunity to mutate. Okay? These guys had the same thing. The only difference was they didn't have Diet Coke. They didn't have something causing mutation. Bang. I've got a very simple answer. So the Ames test allows me to answer very quickly the question, is this compound mutagenic? Does it favor errors in DNA replication? Questions on that? Two questions. Cassandra.
Couldn't something be mutagenic to bacteria and not to humans?
Very good question. So Cassandra says, couldn't you have something that's mutagenic to bacteria and not to humans? Absolutely. You can. Okay? So this wouldn't be the tell-all, see-all. But this would be an indication that perhaps we should do some mice studies, for example.
Have they done human studies?
Okay. Well, doing human studies is difficult. Okay. And the reason human studies is difficult is it's kind of hard to say, hey, I want to inject this compound in you and see if you get a tumor. Okay. So if you get indications here, if you had an indication here, this would immediately pull things back where you wouldn't want to be putting it in food, and then you would do further studies on mice to see, is it happening in an organism that's at least more closely related to humans. So that would be the number one thing that you would do. Okay. You had a question too.
Yeah, is Diet Coke mutagenic?
Is Diet Coke mutagenic? No. Diet Coke is fortunately not mutagenic. Lori.
How do you maintain a bacteria culture that doesn't have a lot of mutation?
Okay. All right. So how do I maintain this culture so that it keeps this plasmid and does its thing? Okay. We're getting kind of techie here, but I'll answer your question. You won't have to know this for the exam. All right. So this plasmid that I have that has this gene that's lacking that one base is grown in a culture that has a different antibiotic in it. Let's say tetracycline. And that plasmid has a functional tetracycline gene on it. So I can select that all the bacteria will have the plasmid by the virtue of the fact they grow in tetracycline. Okay, so I know everybody's got the plasmid. And then the question is, when I put them on ampicillin, how many of them have that mutation? Is that, yeah. Okay, good, you guys think about it. All right, that's the Ames test, and that's how we tell, or one of the ways we tell. There are flaws in this. I meant to mention also in Cassandra's question. We can say, well, something is mutagenic in bacteria but not in humans. What if something is mutagenic in humans but not in bacteria? It won't be picked up by this scan. So there's other tests that will be done. This is a very quick and simple type of scan. And the other thing you might imagine is a problem is, what if this compound in Diet Coke can't make it into bacterial cells? Right? Then you'd never know. So this is not the only way that we look at mutation. There are other ways of studying mutation. In fact, what you can do, you can imagine a test where I took a DNA polymerase and I used the DNA polymerase with a bare stretch of DNA and I allowed it to replicate and asked the question, how many mutations happen here? Then you avoid the problem of getting it into the cell, etc. So other things like that are done to test. Okay. The last thing I want to talk about is recombination. And recombination is something I think we just sort of take for granted. All right? Something we take for granted. Recombination happens for a variety of reasons. One of the most common reasons that it happens is because related sequences of DNAs in different chromosomes align with each other. And when they align, there are enzymes that can basically swap portions of them. And that's what's happening in recombination. So we see schematically here that we've got this red-blue pair. And now we see that a portion of this red-blue has gotten an integration of a brown that's here. All right? So we've moved a portion that might be both strands or one strand. It doesn't really matter for our purposes here. But we've moved a portion of it from one strand to the other. Are these going to make perfect base pairs if these were making perfect base pairs? Probably not. There's probably going to be some mispairing in there, which will get rectified during DNA replication. Okay? And no, it won't go back to the original parent necessarily. So we're going to see some changes happening as a result of that recombination. And that recombination is happening all the time. And it happened between the time that the egg that you grew from, the fertilized egg that you grew from, and the time that here you are now. Meaning that we always like to think of, well, I got one set of genes from mom, and I got one set of genes from dad. But in fact, I have a set of genes that's neither mom nor dad, but actually a combination of the two. I tell you that so that you know that you really are unique. You're not just mom and dad, you're somebody who's really special. Okay? You've got DNA that neither one of your parents had because recombination has happened during that time. That's kind of a pleasant thought, isn't it? All right. Well, how does recombination happen? You could teach a whole course on recombination, and believe me, I'm not going to do that. It is a fascinating process. There are different schemes that are involved, and I'm just going to throw some names out at you just so that you're aware of them. One is called Strand Invasion. Okay? We see a system here where we have these similar sequences that are aligned. There are enzymes that will peel off a strand and basically insert it in, as we can see here, thereby displacing the other strand. Once that has happened, then there are other enzymes that will basically clip off the excess, and you've got your integrated DNA. One of the things that those systems give rise to are what are called Holliday Junctions. Okay? Holliday was the name of the person who discovered that, and Holliday is spelled, in his case, with 2 L's in his name, not 1, as in holiday, like Christmas. Okay? Holliday Junctions, here is a set of 2 DNAs. Here's 2 DNAs all by themselves, right? Here are 2 DNAs undergoing this invasion, and we see that they form what's called a cruciform structure. From the side, it looks like a cross. Okay? It's a cruciform structure, and this cruciform structure can twist and untwist, and we allow this strand to move a certain distance. That's part of this isomerization process that's happening here. Then we have 2 ways this guy can be resolved. We can cut it across this way, or we can cut it across this way. Okay? Depending upon how we cross that, we're going to make some unusually recombined DNAs there. And recombination gives rise to things that you've already heard about, some of which are bad. Anybody remember BCR-ABL from last term? BCR-ABL, which had a fusion between the BCR gene and the ABL gene, that happens because of recombination. Okay? Recombination has benefits, just like it has detriments, just like mutation has benefits as well as detriments. Okay? Recombination is, unfortunately, a fact of life. You're not going to change it. It's happening in your cells right now, potentially giving rise to something that's going to kill you, or potentially giving some of your cells a better ability to function, like hopefully in your brain before Friday. Okay? In any event, once this system gets resolved, then we have new sequences that have been paired with each other. One of the things that's critical in the functioning of this is the action of enzymes known as recombinases. A recombinase. Recombinases facilitate this integration of sequences from one strand into the other. And they have to do some very important things along that way. One of the things they have to do is make sure the tangles aren't too big. You start putting a strand into a duplex, you're going to have some unusual possible structures that are going to arise. And not surprisingly, what happens is when we look at the structure of recombinases, we see in many cases they look very much like topoisomerases. Topoisomerases, of course, are involved in untangling things, and topoisomerases themselves may play roles in certain types of recombination. When we look at HIV, for example, one of the things about HIV that is critical for its life cycle is an enzyme called integrase. Integrase allows the HIV DNA that has been made as a result of action of reverse transcriptase. It allows the DNA to be integrated into the host chromosome. So here's a double-stranded DNA, and all of a sudden it gets inserted into the host chromosome. That is a recombinational-like event, and that event requires an HIV enzyme called integrase. And integrase looks an awful lot like a recombinase, which looks an awful lot like a topoisomerase. Okay. Questions on that? Laura?
Can you only have recombination between same chromosomes, like pairs of chromosome 1?
No.
Or you get like chromosome 1 and
Yeah. So the question is, can you only have recombination between the same chromosomes? And the answer is no. Can you restrict recombination only to sequences that are related to each other? Okay? It's very easy to see how aligning these things might cause things to cross over. And there are certain recombinational schemes that appear to have no relation to each other where they integrate. Integrase in HIV is a prime example. HIV appears to integrate almost randomly in the genome. It's not going into homologous regions. Most recombination is, there is some relation that you can see, but not all recombination involves related sequences that you can see. Yes, Megan.
So how frequently does this usually occur? I mean, is this happening all the time?
It's happening all the time, but it's a relatively infrequent event. Okay, so if you're asking how frequently would it happen with respect to cell division, I don't know. It will depend a lot on the environment of the cell, what the stress conditions that are in, and so forth. But it's not something that's happening overly frequently. It's not happening. It's not something you're going to sit and worry about a lot in terms of making that BCR-able fusion, for example. But it does happen. Okay, that's a good stopping point for that. We've got about 10 minutes. If you'd like, I will be happy to take questions in anticipation of the exam. And if not, we'll just finish early. So it's up to you. Other questions? Yes, sir.
Can you just go over the important parts of the structure of B and Z DNA?
Yeah, B and Z DNA. So I'd be happy to. So if we look at the different types of DNA, what we see, this is just a table, as I said. I'm not concerned that you memorize anything in this table. Looking at B DNA and Z DNA in simple terms, number one, B DNA is present in about 99% of all the structure of DNA in your cell. Most of the structure is in the B form. Okay? Z is relatively infrequent. I shouldn't say relative, it's quite infrequent, but it's there. We know it's there. Structurally, there are two primary differences. Okay? One is that the B DNA has what we describe as a right-handed helix, describing the way in which the strands are wound around each other. Okay? Z DNA is backwards. It has a left-handed helix. Okay? So that's a very big structural difference. Both use the same base pairing system. G with C, A with T. That's good. Okay? The other primary difference between them is that Z DNA is elongated compared to B DNA. Okay? So if I had, for example, something in the B form, I might have 10.5 base pairs per turn. If I had Z DNA, okay, then I might have 8.5 base pairs per turn. That thing is stretched way out. Okay? So fewer base pairs per turn means that the bases are further away from each other on each one. Okay? That's the two biggest structural differences that you see between them. Okay? Yes, Megan.
So you were saying that fatty acids that are larger than 16 carbons long get broken down in the peroxisome, or
Okay.
oxidized in the peroxisome?
That's correct. Uh-huh.
So the electrons and protons get used specifically for making hydrogen peroxide, and so
Yes.
you can't use the energy from 16 carbons.
Okay, so her question is, I said that fatty acids longer than 16 are broken down in peroxisomes. There's no electron transport system there, and so we're donating those electrons and protons to oxygen to make hydrogen peroxide. Her question is, does that mean we don't get energy out of fatty acids above about 16? And the answer is no. Because in the process of doing this, we are still breaking off and making acetyl-CoAs. The acetyl-CoAs themselves can go out of the peroxisome to the citric acid cycle and be oxidized. So yes, we are still getting energy out of them. We're not getting as much energy out of them as if that oxidation were going on in the mitochondria. Make sense? Yes, Ryan.
So purine and pyrimidine synthesis both uses PRP, but only one of them uses PRP aminotransferase?
Okay, so his question is both purine and pyrimidine nucleotide synthesis uses PRPP. Only one uses PRPP aminotransferase, and that's correct. That's the purines. And the reason for that is that the aminotransferase is catalyzing a specific reaction. That's that first step where that amido group is put onto the ribose, onto the PRPP to make that. In the case of pyrimidine biosynthesis, what's happening is the ring, you remember, is made first, and then the ribose is added. So it's a different reaction that's happening. So there's only one that uses the PRPP aminotransferase. Yes, Jessica.
You were just talking about the B and C differences, and your table shows that the 12 base pairs per turn is the same.
Yeah, That's not a very good thing to say, was it? The turns are further. Okay, so I said 8.5, and that's not correct. Okay, but to make a turn, it has to go a further distance. The repeat distance that you see, where's the repeat distance? Pitch per turn of helix. So they're stretched further out. 8.5 is not the right number. Okay, but they are stretched further out before you get a full turn. So I apologize. Yeah.
And then the A is more compressed in comparison.
A is more compressed compared to both of them. That's correct. And so the fatness of these varies as well. So this guy is the thinnest, this is the fattest, and this is the in between. Other questions? Yes?
The electrons for desaturases, are they going to FADH2 or NADH?
Okay, so for the desaturases, which enzymes? The desaturases?
Yeah.
Okay. So when we look at what's happening with desaturases, they are, let me think about that. So in the case of desaturases, it's a complicated scheme that they use. And so ultimately what's happening is the electrons are coming from NADPH and getting donated to oxygen. So I think that's that figure I showed you, if I can pull it up here, that actually has a fairly complicated scheme for the movement of the electrons. And make sure we're talking about the same thing. Yep. Okay, so making double bonds. This figures it. We're talking about the same thing here? Okay. So I said NADPH, it's NADH. But this is a very complicated scheme in that we're actually starting with electrons being donated. And what's happening in the process is they are sucking electrons this direction. Okay? So ultimately, and it's not even shown on here, ultimately they're going to oxygen because they're going into the electron transport system, just like they would otherwise. But neither one of these is an end product of that. This, as I say, it's a complicated scheme. But the electrons are moving this direction, and from here they can actually go into either one of these and end up in the electron transport system. Okay? Everybody's frowning. Thinking about the exam. Other questions? You guys all ready? No? Okay. All right, hang in there. Hi, yeah, what's up?
Clarification of your notes. I think I just got confused. But in talking about gout and so gout is often accumulates in big toe. Uric acid acts as an antioxidant and is protective against multiple sclerosis. And it says the disease is successfully treated with allopurinol.
So the disease is gout. Yeah, yeah.
So technically, if you were taking allopurinol, you might be more susceptible to MS then.
In theory, you would argue you might be. Yeah.
Okay, I just want to make sure I have that.
Yep, you got it.
Okay. I'm missing my hand, but if you were to clip the Z form, would it relieve the tension to make terminal-based space particularly? The terminal series, or?
Well, it's not quite that simple. So what's happening in the Z form is the Z form happens as a result partly of superhelical tension and partly of sequence specificity. Okay, so it's not simply the tension that's the factor.
So it's technically relaxed.
Yeah, the Z form has its own relaxed state. Yeah.
Hey, I had a quick question. So you do that interview course in the Honors College every quarter?
No, I do it fall and spring.
Oh, okay. Okay.
Yeah.
So spring, I'm not in the Honors College. They'll have preference, right?